The overall goal of this methodology is to assess the susceptibility of influenza A and B viruses to the neuraminidase inhibitors by using a fluorescence-based assay.
This method can help determine whether an influenza virus is resistant or sensitive to the neuraminidase inhibitor class of antivirals. The main principle of the assay is to see whether the neuraminidase inhibitor antiviral blocks the action of the neuraminidase enzyme activity.
Prior to antiviral susceptibility testing, the virus must be grown to high viral titer by either passaging in cell culture such as the MDCK cells or in embryonated chicken eggs. In this demonstration, I will be using live influenza A viruses of the H1N1 and H3N2 subtypes.
Beginning with a 96-well U-bottom plate, dispense 120 microliters per well of undiluted cultured influenza viruses into column 1, and 60 microliters of 1 times assay buffer containing 0.1% NP40 into the remaining 11 columns. Using a multi-channel pipette, serially perform twofold dilutions across the plate, leaving column 12 as a blank containing only 1 times assay buffer. Transfer 50 microliters from each of the wells into a clear 96-well flat-bottom plate. Add 50 microliters of 300 micromolar Munana per well, and gently tap the plate to mix. Then use a plate sealer to cover the plate to prevent evaporation, and incubate the plate at 37 degrees Celsius for 1 hour. Add 100 microliters of stop solution per well to terminate the reaction.